the cellular profile, about the abilities and potential to detect as many subpopulation of cancer cells that we have in a cancer, in a tumor, detection of subpopulation, and detection and isolate of minimum residual disease. What kind of novel techniques we are talking about? We are talking about flow cytometry techniques, which actually is single cell detection and interrogation technique. We are talking about real-time PCR or quantitative PCR, a very sensitive method. We will talk later about this. Microarrays, which actually can be performed and provide us information concerning the gene expression profile from the old genome, or it can provide what we call CGH, comparative genome hybridization, and provide us information concerning the mutation that may exist inside cancer cells' DNA. Valuable information that can be translated easily to abilities of our cancer cells. Flow cytometry. We put either a tissue sample which can be dissociated in a single cell suspension, or a blood sample if we want to detect what we call circulated tumor cells, and they can pass through a laser beam which interrogates our cell, which previously may have been stained by specific antibodies and fluorescent dyes. So we may have information in this dot plot or histogram form and detect even the tiniest cell that may exist on our sample with specific feature of our need. Of course, it sounds very simple and easy. It's not, because it needs a lot of experience, needs a lot of work, and much more standardization of the method. As I said, it's a single cell analysis. It can provide information with forwards and side scattering concerns the size and granularity. And by the staining, you can see antigens. Remember, genetic is very important, but it's not the target. The drugs actually inhibit or target proteins, antigens. So genes frequently, or most of the time, are in linear relation with the antigens, but not always. So we must keep that in mind. Also, it can provide flow cytometry information about the DNA quantity and quality. We can perform DNA index analysis through flow cytometry and see what happens with our cancer cells. And of course, we can perform viability and apoptosis detection with several kits like an XNV or PI, etc. Very powerful technique, very useful, and very fast. Quantitative PCR. It's actually the detection of fluorescence when we perform a PCR, an amplification of a gene. In case of cancer cells, we isolate the RNA. We perform quantitative RT-PCR. So we isolate the RNA, we perform cDNA synthesis, so we have a double-stranded DNA, and then we amplified it. And there are specific dyes who actually bond with this DNA and start bleach and give us signal. And there is a crucial term called cycle threshold, which means in which cycle we may start have signal. And there are controls which can give us numbers of and quantity of specific antigen, which can also translate it to number of cells. Equally important information if we are talking about the circulated tumor cell number. The microarray, which means that it's an hybridization actually technique which performed in very high dense slide with probes which are immobilized up on this slide. But first you need to isolate the cells. That's why we talked about flow cytometry and sorting. The reason is that when you interrogate the cells and identify the population of your interest, you may isolate them here or here, because the flow cytometer can chart every drop and every cell of your interest, and according to this electrical field deviate from the drop of waste. And this is an isolation technique. Remember that, because isolation is completely different from what we call enrichment. When you have the cells, nowadays, very frequent I receive the question, how it's possible for few cells to perform microarray analysis or to perform all these profile. So any man on our field knows that nowadays, the last 3, 5, 6 years before, there is the technology to do a single cell diagnosis. Right now, there are companies like Advalytics who perform real-time PCR on a grid of a single cell suspension. So it's not something which is far away or highly trained technique. It's simple tests and simple assays that exist in the market. If someone has the question, simply search. It's out there. So when you perform a cDNA synthesis, actually you can hybridize your cDNA, which can come from your RNA, almost to whole genome. Nowadays, we use in our laboratory the Inopsys 740. And we use slide from Combi Matrix, which has 90,000 dots, 2.5 times more than the human genome. Easy and simple. So you can hybridize your cDNA and see information which is important for how you will deal with your cancer. For example, DHFR, TS, or Topo 1 or 2 are targets for cytotoxic drugs.